All right. We're gonna start some neurology. It's gonna be great. There's 3 PowerPoints for neuro. I only had one originally online. I just got the other 2 up. So there's an intro to the nervous system, which we're gonna start in first. We're gonna spend like 20-ish minutes there. Then we're gonna do the nervous system part 2. We're really gonna just look at action potential and neurotransmission in that PowerPoint. And then the third one, we'll spend some time looking at the autonomic nervous system. So that's really what we're going to go through here. But this first one, I really want us to start with this intro to the nervous system because we can review neuroglial cells or microglial cells, which are tested on your NPLEX. And I feel like we forget about them if we can. So we're going to review them. All right. So we're not going to go through some of these basic pieces. It is helpful to kind of go through your overview of the nervous systems, like how things are broken down. So CNS, your central nervous system, your brain, and your spinal cord, your integration and control center of the body. Your peripheral nervous system is going to be anything that's outside of your central nervous system. So everything else, including spinal nerves and cranial nerves. Of your peripheral nervous system, you have sensory and motor division, sensory afferent, motor efferent. So sensory is stuff that you're going to be sensing, it's bringing it back. Efferent exits the brain, right? Efferent, E and E, they exit the brain. It's going to be from the brain outward to the body. So our sensory afferent division, we have somatic and visceral sensory fibers. Somatic is conveying impulses from your skin, muscles, joints, as well as any related tissues to those areas back to the central nervous system, versus visceral is really your organs. So what are your organs feeling? Those sensations. The motor or efferent division is transporting impulses from your central nervous system to effector organs, organs that they affect, right? That's all that means. There's 2 divisions of our motor system, the somatic and the autonomic nervous system. Somatic is your contraction, your conscious control of your skeletal muscle. Autonomic is everything that's autonomic that we don't consciously control. It is involuntary, and it's our sympathetic and parasympathetic nervous system, which we will look at those 2 here at the end. So if you wanted some organization, I think this is probably your best summary slide. Oh, yeah. Oh, thank you. Sorry, online people. Thanks. Hi, hello. I'm the problem. Okay. There you go. Can you see it now? Yes. They can. Awesome. Thanks, Claire. Thank you. All right. Perfect. All right. So this is our big summary slide of our nervous system. So if you know this, you're good for at least the divisions. There'll be a couple questions about divisions of the nervous system. They'll really try to trick you with the afferent, efferent conversation. So calling sensory afferent, motor efferent. They'll play back and forth with the somatic nervous system, the autonomic nervous system, like sympathetic and parasympathetic. I think those questions are relatively easy to answer. So I think more or less it's the afferent, efferent discussion. Okay. The main reason I wanted to do this is to talk about neuroglial cells. Who remembers neuroglial cells? Some people-ish, kind of harkening back to neurology in the fall. So they're any cells that's not neurons, right? They're all of our neuron support cells. So we can separate them into central nervous system and peripheral, and this is often really great test questions because they are different. So in our central nervous system, we have our astrocytes, our microglial, our ependymal, and our oligodendrocytes. These are the 4 types of neuroglial or support cells that support neurons. Our astrocytes are the most abundant. That's been asked before. What's the most abundant neuroglial cell? Astrocytes. They are going to do really every function that you can think of. They are the most diverse in function. They have actual structural support to neurons. They will help with capillary, so blood exchange for neurons, providing them with nutrition or optimal operating conditions. They'll help with migration of neurons to their resting place or their adult working place. Because they can interface with capillaries, they help to control the chemical environment surrounding. They help with neuro response from neurotransmitters, and they can help with information processing. So what do astrocytes not do? They pretty much do everything except for anything with myelin, and they don't really do anything with defense. So they do everything else, but they're not defending neurons from attack, and they're not really doing anything with myelin production. But astrocytes really have big roles in supporting neurons, and they're the most abundant neuroglial cells in the central nervous system, right? We're talking about in the brain and spinal cord. Microglial cells are going to be your immune cells of the neuroglial cells. They're going to go towards injured neurons. They can become phagocytes. They can phagocytize microorganisms. They can also clean up debris from any damage or attack. And they can help essentially defend or heal a neuron that's damaged or injured in the central nervous system. Ependymal cells are your kind of part of your blood-brain barrier. They're lining these fluid-filled ventricles. They're lining the cavities of that contain cerebrospinal fluid, and that makes sense because they have cilia. The cilia will beat to allow for circulation of cerebrospinal fluid. So how is cerebrospinal fluid circulated? Partially due to your ependymal cells that are lining these ventricles. So it's again another great question, kind of combining those two knowledge areas. They help create your permeable barrier between your CSF as well as your brain. So they're creating that barrier between the two. They're not providing nutritional support. They're not defending from anything. They're really creating a barrier, and they're helping circulate CSF in the central nervous system only. And then lastly, your oligodendrocytes. These are the myelin producing and myelin supporting and myelin creating cells of the central nervous system, not the peripheral nervous system. So if we talk about a condition that causes CNS myelin damage, these are the cells that potentially are being attacked or potentially being affected. An interesting piece with the oligodendrocytes is you'll see one cell body and multiple myelin sheets attaching to multiple axons. So one cell can provide myelination to multiple axons or multiple neurons, which is unique. And again, these are only in the central nervous system. Okay, so 4 types. 2 types in the peripheral, satellite and Schwann cells. Satellite cells, you could see, are the astrocytes of the peripheral nervous system. So satellite cells are neuroglial cells in the peripheral nervous system, and their function is like an astrocyte. So they do all the things except for defense, and they don't do anything with myelin. Schwann cells are your myelin producers, myelin supporters of the peripheral nervous system. So Schwann cells are like your oligodendrocytes. The difference is that Schwann cells, you have one cell for one axon or one neuron. The Schwann cells are going to wrap around almost like a jelly roll. They kind of like roll themselves around the axon itself. And so there's only one Schwann cell that's ever affecting one space on an axon at a time. So you'll have multiple Schwann cells covering the whole length of the axon peripherally. This is super important for regeneration of peripheral nerves. So if you have a crush or cut injury, your Schwann cells are going to come in. They're going to try to help pull together the 2 ends of the axon and help those cells repair. So they do have some repair function. Okay. So 6 cells, 4 central, 2 peripheral. If you know that enough, that kind of base knowledge, you'll be fine for those questions. Okay. Neuron, the only pieces of the neuron I want you to be able to recognize is your neuron cell body, often called the soma. So they will use that. They don't use perikaryon, but soma, they will refer to a neuron cell body as soma sometimes. And it's also the biosynthetic center. So where the cell body is where all the biology is. It's where all the organelles are. It's where the production of everything happens. The axon is just the travel highway where stuff is taken down, either physically or the electrical signal is sent down. But no actual production is happening in the axon. It happens all in the cell body. There can be storage at the axon terminus, and there can be some transformation that happens in vesicles for neurotransmitters. We'll see that. But there's no actual like biosynthesis taking place down the axon or in the axon terminus. It's all happening in the cell body. The neuron cell body, you'll have most of them, the majority of neuron cell bodies, not all, but most are located in your central nervous system. Nuclei are clusters of neuron cell bodies in the central nervous system. So that's confusing, right? Because nuclei has lots of other meanings too. But if you're seeing a question asking about cluster of neurons in your CNS, not in your peripheral nervous system, you would call it a nuclei. If you see a cluster of neurons in the peripheral nervous system, you call that a ganglia or ganglion. Okay, so you'll see those words used. There's been NPLEX questions on this pretty consistently because it's a really great way to trick you. So nuclei is a cluster of neuron cell bodies in the central nervous system. Ganglia as clusters of neuron cell bodies in the peripheral nervous system. You could also see them as nucleus or ganglion. Same thing. Questions on that one? Okay. I see some chat. Oh, awesome. Thank you. When it comes to neuron processes, they're arm-like components. There's dendrites and axons. I don't find them testing too much on these components, but we'll just look at the differences. Dendrites, you could have multiple hundreds of dendrites. It's the input region of your neuron. You could think of it as like the feelers that are going out to kind of see like an antenna of the neuron to kind of see the environment, see if there's any incoming signals, any local or graded potentials that it's going to need to respond to. It's going to convey any incoming messages that are happening in the localized area. It's not going to be sending messages out. That's the axon's job. The axon, each neuron you could say has one axon, but that's not completely true. There can be some that have more than one, but traditionally each neuron has one axon. Axon is the highway, the information highway that your action potential is sent down. You can have, though, because it is a highway, it's a physical structure, it's made of microtubules. You can have actual substances moved down the axon via transport proteins, the little cute proteins that look like they're walking along microtubules and pulling something. I don't know if you've ever seen that video, like my favorite cell type, or organelle. But movement down an axon when it comes to moving physical stuff, not an action potential, but physical items like a neurotransmitter or proteins or waste can go both directions. When you're talking about an action potential, the electrical signaling, that's unidirectional. So this is a great test question as well, because we're talking about two different things. So if we're talking about movement of stuff, movement of proteins, movement of neurotransmitters, both directions down a axon. Away from the cell body is called anterograde movement. Retrograde movement is towards the cell body. So if I am a neuron and I have created a neurotransmitter and I'm going to send it down my axon highway, and I'm now the vesicle walking away from my neuron cell body, I am moving anterograde away. Now, if I'm at the end, if I'm a vesicle and I have waste products from the breakdown of whatever neurotransmitter I used, and I want to bring it back to the nucleus for digestion, the actual nuclei and cell body, I'm going to walk this way back towards my cell body, which is retrograde movement. If I'm an action potential, if I'm an electrical signal, I can only go from cell body to the terminus. I can't go backward. Okay. Right. That's a good test question. Myelin, I mentioned it already. Myelin is just protein lipid structures. They're protecting, they're insulating. You could think about it as electrical tape along a copper wire, right? It's going to increase the speed of signal, of the electrical signal down an axon, going to make the signal be sent more efficiently or effectively. We can see here, this is an example of the Schwann cell, like I mentioned, with kind of its jelly roll appearance. In between each myelin place down an axon, there will be gaps. The gaps can occur between Schwann cells and between oligodendrocytes. These gaps are where there's exposed axon to the environment. This is where we'll see all of the regeneration of the action potential when we start talking about that process of propagation. Underneath myelin, we don't actually have to do that work. The signal's just sent. In the spaces between myelin, that's when we're going to see these different ion channels opening and closing, and these gaps between the myelin sheath. I don't hear them called nodes of Ranvier. In a non-myelinated fiber, it's having to do this process of opening and closing channels the whole way down the axon because there's nothing that's covered. Hence why it's a little bit slower to transmit a signal, because those ion channels are having to open and close at every point along the axon, versus if they're covered in myelin, they only have to do that during these gaps in myelin. Otherwise, they can just kind of transmit down, go down their superhighway. Okay. Great. The only other component here about myelin that I'll mention is when you're looking at white versus gray matter. White matter is typically regions of the brain and spinal cord where you have lots of myelinated fibers and less neuron cell bodies. Neuron cell bodies look more gray, they look a little bit darker because of all the organelles. So in the gray matter, that's when you're going to see your cell bodies and non-myelinated fibers. So if you think about loss of gray matter versus loss of white matter, you could kind of think about what is being affected there. Loss of gray matter, you're losing neuron cell bodies. Neurons are lifelong, so if you lose them, you don't get them back, right? We don't regenerate neurons. We could regenerate part of an axon, but we don't regenerate a neuron cell body. So if we lose gray matter, if we're actually losing neuron cell bodies, that's significant, going to have more significant potential pathology. If we lose white matter, that's myelinated axons. It's still not great, less efficient signaling being sent, but you still could have maybe an unmyelinated axon that's sending a signal, okay? So that's our different discussions. You can kind of lean into that without even really knowing the pathology, without even knowing the condition. If it's talking about gray matter, it's cell bodies that are potentially being lost or damaged. If it's white matter, it's myelinated axons for the most part in the brain and the central nervous system. Okay. I don't see types of neurons tested on that much, so I don't think it's high yield. I do have it here if you want to review. It really comes to the definition of how many processes are located on the neuron, meaning are there multiple axons, are there multiple dendrites, how much stuff is coming off the neuron? I see this rarely tested on, so I'm going to skip it, but if you want to review it, it is here. One thing that can be asked that people forget is an interneuron. Like, what the heck is an interneuron? It's an association neuron or like a communicatory neuron. It's typically lying between motor and sensory neurons to help them communicate with each other. The majority of interneurons are in the central nervous system. You'll see a lot in the spinal cord itself, helping transmit pathways from sensory to motor. 99% of the body's neurons are interneurons. So if they asked you what's the most common type of neuron, and interneuron was an option, that would be your answer. And that has been on the test before. Otherwise, I wouldn't learn the others necessarily. Okay. It's a brief question to see kind of where our brains are at. Nervous system has 3 overlapping functions. Which of the following represents the logical sequence of these 3 functions? So we're looking at sensory and motor input and integration. I'll give you a second to think about it. Yep, so sensory is first, we get a stimuli, then we integrate, and then we respond. Good. So just seeing if we have any recall of our autonomics, or our sympathetics, or our afferents, or our somatics. So when you study for an exam and you reach for a beverage, you extend your arm, what do you activate? Which part of your nervous system? Somatic, right? Because you're actually reaching, you're doing a conscious muscle action, so somatic. Door slams, you flinch, your heart is racing forcefully. What part of your nervous system responded? Autonomic, great. Electrical wire to electrical insulating tape is peripheral neurons to what type of cell? Schwann cells. Remember, Schwanns are the insulators, the myelin producers peripherally. Oligodendrocytes are the same centrally, so central nervous system. So if I ask the same thing for central neurons, it'd be oligodendrocyte. How long do neurons live? What did you say? Huh? D, this whole time, lifelong, forever, until they die. Yep, your lifespan. Which is why we want to keep them. Movement towards the cell body of a neuron. A, retrograde. Yep. Axon portion that communicates with its target cell. Ooh, good. The terminus. Yep, good. Basis for differentiation between gray and white matter is presence of what in white matter? Myelination. Yep, good. If I said the presence of what in gray matter, I'd probably say neurons would be, or cell bodies would be the best answer if I was asking about gray matter. Most common neuron, oh, we didn't talk about it. That's fine. Multipolar. Okay. Okay, great. That was our little cellular look back into the world. We're going to do action potential first, and then we'll talk about the autonomic nervous system because that's more fun. I love action potentials, but not a lot of people like them as much as I do. Okay. Why do I want you to know this? Because it's going to be asked of you in some way, shape, or form. All right. Whether it's in the nervous system or the cardiac system, the musculoskeletal system, they're going to ask you a question on it. And so I want us to at least have some general understanding of what cells are involved. So action potentials are not decaying over time. It's an all or nothing event. It's either going to happen or not happen. Okay. In neurons, an action potential is often referred to as our nerve impulse. We're going to be talking about voltage-gated channels. So our key players are going to be our sodium voltage-gated channel and our potassium voltage-gated channel. Potassium voltage-gated channel is easy. It's either closed, no potassium can get through, or open. Potassium can get through. Sodium voltage-gated channels is either closed, where no sodium can get through, open, where sodium can get through, or inactivated, where sodium's temporarily blocked by this secondary activation gate. This activation gate, this allows the sodium channels to be opened and closed a little bit more rapidly than potassium channels. So we get a little bit more overarching control over sodium because of this inactivation gate that can temporarily block the channel for us. This then process is reset back to closed before we start over again. So we go closed, open, inactivated, and then back to closed. That's the process of sodium. Potassium, closed or open, easy peasy. Okay, so these are the players I'll refer to. So when I'm saying activation gate, I'm talking about actually the main tube, the main channel, the main pore. When I say the inactivation gate, I'm saying the thing that blocks the channel or blocks the pore. So if we have an active inactivation gate, that means the channel's being blocked. If I have an inactive inactivation gate or a closed inactivation gate, that means the inactivation gate is at its baseline. Okay. I realize how that's confusing. That's why I wanted to show the picture. It's like, what the heck are we talking about? But this is our body. This is what we have to work for or work with. Okay, action potential 4 steps. Let's get through it. Resting state, everything's closed. Sodium and potassium gated channels are closed. There's leaky channels for sodium and potassium open. That's allowing sodium and potassium to flow down their concentration gradient across the plasma membrane. Inside of the cell, what ion tends to be more concentrated at resting state? Potassium, and therefore what is more concentrated extracellularly? Sodium, that's where it likes to live. Is inside the cell more positive or negative than outside the cell? More negative. Inside the cell is more negative than outside of the cell. So that's our baseline resting state. We have more potassium inside, more sodium outside. We're having ions moving through leaky channels through passive diffusion down their concentration gradient. That doesn't stop, that happens constantly. These aren't blocked or affected. And inside the cell is going to be more negative than outside the cell. That's our baseline. So our sodium channel, we are in its closed state. So our activation gate for our sodium channel is closed. The pore is not letting anything open. The inactivation gate is technically open, right? Because the inactivation gate of our sodium channel here is not blocking anything. So at resting, our activation gate is closed, our inactivation gate is open for sodium channels. For potassium channels, it's closed. Okay, that's our baseline. At our second step, essentially, is depolarization. Depolarization is going to be the increased membrane positivity. So the membrane is going to become more positive, or you could say the difference between inside and outside of the cell is becoming smaller, right? It's getting closer to baseline. It's getting closer to being the same. So our sodium channels in depolarization open. The reason that sodium channels open is due to typically a local or what we call graded potential. a local change where there was some like positivity surrounding that area. That's what our dendrites are sensing. Our dendrites are saying, is there any positive polarity change? Is there any signals being sent anywhere that are causing a graded potential, a slight membrane change that's enough for me to trigger an action potential? If that graded potential is high enough, the dendrite will tell the neuron to fire an action potential. That then neuron will send the action potential down the axon. So that's how this process is started. So that's often a question students ask is like, well, why did the cell decide to depolarize? It's because a graded potential reached threshold. It got positive enough that the membrane said, okay, I'm to this threshold state, it's typically plus 20-ish of resting membrane potential, then now I can fire an axon or fire an action potential and send it down the axon. So if all of that's happened, that's when we get depolarization. Otherwise, we stay at the resting state. So if we've reached threshold, we're going to fire an action potential. It's an all or nothing event. So if it happens, it's happening. If that takes place, we depolarize by our sodium channels open, voltage gated sodium channels open. We have an influx of sodium going into the cell. So a +1 charge going inside of a cell is going to increase the positivity of the cell interior. That's going to increase the actual resting membrane, increase the membrane potential. So we're going to make it more positive. The intracellular fluid becomes more or less negative, and we get this massive membrane spike. Nothing's happening with potassium yet. Okay. After it reaches its natural spike plus 60 above, that's like what the voltage changes to ish. I wouldn't worry about numbers, but once that occurs, it's always going to be at the same height, the same intensity. Then our sodium channel, it's like, well, I don't have the charge drive or the concentration drive to run inward anymore, because now inside the cell, there's a lot of sodium and there's a lot of plus or positive charge. So that drive is going to be less when we're up here. So what happens, the sodium says, well, I don't want to go in there. We need to close this off. We've changed this potential. There's no drive for me to flow inward. So the inactivation gate then closes. It's going to block this channel so no more sodium can come in. When this happens, about the same time, the potassium channel opens and potassium rushes out to help neutralize or rebalance said membrane. So now sodium's inside the cell, the charge is really positive, and potassium is leaving the cell. Potassium is going down its charge and concentration gradient. It's going from an area of higher positive charge to lower positive charge, and it's moving where there's higher concentration of potassium to an area where there's lower concentration of potassium. So it has drive to leave. We call that repolarization. At the end here, so we had stage 1, stage 2, stage 3. There is a stage 4, which is hyperpolarization. This is where we actually go a little bit below resting membrane potential, and this helps prevent an action potential being repropagated too soon. So hyperpolarization prevents us from sending an action potential too close to another action potential. This can be overridden if there's a stimulus that's strong enough, but typically it isn't, and so we have some relative control over how many action potentials are sent down the axon in a given time. Okay, so in this state, sodium channel or potassium channels remain open, so potassium is still leaving down its electrochemical gradient. We now will see the sodium channel having time to reset. So the actual activation gate here gets closed, and the inactivation gate is released and gets open. So no sodium is leaving the cell. So there's still a high concentration of sodium at this stage inside. We still have sodium inside the cell. There's sodium outside. We haven't reset this membrane really yet completely. None of these processes we've discussed so far have allowed sodium to leave and potassium to come back in. We're just trying to rebalance the charge right now. Okay, a little picture. So how do we reset it after we're done? After we go here and then we get to this like hyper state, how do we get back to resting? That's thanks to our sodium potassium pump. Sodium potassium pump uses ATP, so it's ATPase, so it needs energy to reset. So every time we are doing an action potential, we are using energy. And what this will do is going to take 3 sodium molecules and bump them outside, and it'll take 2 potassium molecules and they'll bring them in. It'll recreate that electrochemical gradient for us by allowing more positive charge to leave and to come back in, 3 sodiums out for 2 potassium in, so that's a +1, right? And then we have our potassium moving back where it likes to go, which is inside the cell, and sodium out where it likes to go, outside the cell. Okay. The kicker here is it's all or none. So it's not going to partially happen. Action potentials, if it's going to reach threshold, it will fully fire through the whole process. It'll move all the way down the axon, unless if there's some damage or the axon's cut or something. But in a normal physiologic state, it will fully propagate towards the terminal. Okay. What questions about axons or action potential do we have so far? Yes.
Well, in the beginning of the sodium pump,
Mm-hmm.
if that's because of the graded potential, right? You're saying like, you have like a signal to change that gradient.
Right.
What stops the sodium potassium pump from also working at that time?
Sodium potassium pump will be constantly working to pump sodium out and potassium in response to that change in the gradient. So if there's sodium inside of the cell and there's potassium outside of the cell, the sodium potassium ATPase pump will still be working and moving those ions across the membrane using ATP as energy. So it will do that, but if there's not a lot of sodium inside and potassium outside, if that concentration gradient isn't there, the ATPase pump will have less drive to do that swap. ATPase pump is just resetting back to resting membrane potential. The graded potential is different. Graded potential is from a localized change. It could be another action potential coming in, or it could be a local stimulus of neurons in the brain. That's when we say like neurons are lighting up or firing in the brain. Those are graded potentials that we're seeing. Those are local electrical stimulus that we're seeing in the brain that then, if they're large enough, could actually stimulate an all or nothing action potential event. Maybe? Okay. What else? Where are test questions going to come from this? Asking about what ions are involved, where they'd like to be, where they are during repolarization, depolarization, hyperpolarization. Asking about ATPase potentially, like what is their energy needed, or what type of pump resets the membrane, or what is involved in resetting the membrane. All or nothing event questions like action potential. They'll try to get you to say an action potential could have a different intensity. An action potential is always going to be the same intensity, but it can occur more frequent or less frequent. So how an action potential is stronger is the body will send it faster, more of action potentials, more of that same stimulus in a shorter amount of time. If it's weaker, it'll send less of them, but it'll always be the same intensity, the same height or grade. Yes, same thing. ATPase and sodium potassium pump are the same. Sodium potassium pump is an ATPase pump, so you could say they're the same. You have other ATPases that do other exchanges. This is a sodium potassium pump using ATP. Excellent. The only other component here that I haven't really discussed is kind of this propagation piece. But essentially, a question that's often asked or that we think about is, well, why is an action potential going always in one direction? If it's changing the charge somewhere, why couldn't it also go backwards? And the reason is because once you've generated the action potential here, the red zone is where the action potential is peaked at its height, and the yellow is where it's resting, and then the gray is where it's hyperpolarized in that kind of fourth stage. And so if something's hyperpolarized, the action potential, that graded potential is not going to actually affect the hyperpolarized area, because it's going to take a lot more to get that membrane back to threshold. So you actually would need a stronger stimulus to actually cause a hyperpolarized area to propagate with an action potential. That's why action potentials only move one direction. It's not because there's something special saying that they can't go backward. It's just because once an action potential has been sent down an axon, that hyperpolarized area right next door will not be able to regenerate an action potential going back towards the neuron. So it can't travel that direction. There are neurons that are bipolar, meaning they have one cell body, a stalk, and an axon going in both directions. So in that instance, you could have an action potential that started at the cell body and is propagated in both ways, but it's not going to also go back at the same time. Okay, it's always going to move in one direction the whole way. It's not going to kind of bounce back and forth. Okay. Any period that you cannot, or it's harder to generate a new action potential, you could call a refractory period. So we have an absolute refractory and a relative refractory period. The absolute refractory period is essentially during the action potential. So while an action potential is happening, you cannot generate another action potential because you are not at resting membrane. You are already depolarizing and repolarizing. So that membrane itself cannot reach threshold again once it's already reached threshold. It's an all or nothing event. So absolute refractory period cannot be overridden.
Oh, no.
Oh,
Check your
I know.
phone.
terrible. All right. The relative refractory period. I appreciate it. I'm sure that was not in response to this, but the relative refractory period was perfect though. It's like, oh no, it can't be generated. I love that. I hope that was recorded. The relative refractory period can be overridden with a strong enough stimulus. So if a stimulus was strong enough, meaning if it was sent frequent, or if the graded potential was great enough, the local change to the membrane was great enough, you could potentially get back to threshold and generate a new action potential in this relative refractory period. It would just be much more challenging. So it's highly unlikely, but it can occur. And we could see this occur in pathological states. So that's where you can start seeing potentially some of these things change. We're talking mostly physiology today. I mentioned a little path, but for the most part, in normal healthy states, both refractory periods, you're not going to generate a new action potential. Okay. The speed at which it goes varies based off of how myelinated it is and how big the axon is. Okay. The larger the fiber, the less resistance to local change. So the faster the impulse or action potential will be sent. So if it's a bigger fiber, bigger nerve fiber, action potential goes faster. If it's a smaller nerve fiber, action potential goes slower. If it's more myelinated, action potential goes faster. If it's less myelinated, action potential goes slower. And you can have both combinations there. This is just talking about what I mentioned before. When you have myelination, these areas between, you don't have to regenerate that action potential or do any of those channel opening and closing. So you can move through potentially, oh, well, this is there, this one. You can move through faster. You only have to do the stimulus here, and then here, and then here, because this whole area is protected by your myelin sheath. All right. How are we feeling? Can we get through the synapse and then break? Okay, great. So synapse is essentially the communication point between neurons or between a neuron and its effector organ or its target tissue or its target cell. So in the synapse, we have a presynaptic and postsynaptic neuron. Presynaptic means the neuron is moving something towards the synapse. Synapse is the communication point. The postsynaptic neuron is receiving that said information, okay? It's anything after the synapse. So when we hear these words thrown around, we hear it as preganglionic and postganglionic, right? In the autonomic nervous system. Our preganglionic neuron is the neuron that's presynaptic, the one that's sending information to the ganglia just being a cluster of neuron cell bodies. The postganglionic or postsynaptic neuron or neurons are the ones that receive that information and transmit it to target tissue or effector organs. So you'll hear both these terms used and communicated to, and it's telling you what's happening before and after, who's sending the information this time and who's receiving the information this time. You can have synaptic connections between multiple different things. We're primarily going to talk about synaptic connections between two neurons and between a neuron and a cell. Those are the type we're going to discuss. But know that you can have neurons that communicate to dendrites and other things as well. The synapses are chemical and electric. We're primarily going to talk about chemical synapses using neurotransmitters. You can have an electrical synapse as well. It's more rare. Okay. Great. Okay. So to transfer stuff across the synapse, it takes 6 steps. The action potential has now arrived at the end of our axon, and this is the axon terminus. This is our postsynaptic neuron here. So this is the actual cell body. So we could pretend like the nucleus is somewhere like over here. Okay, this is the cell body and then there's an axon going elsewhere. So when the stimulus, the action potential reaches the axon terminus, that is step 1. The step 2 is going to be once that voltage gated change reaches the terminus, it's going to stimulate calcium to move intracellularly from the extracellular fluid to the intracellular fluid. So we have voltage gated calcium channels. When they identify that the membrane has become depolarized from the action potential, they're like, okay, this is my time. I'm going to send in calcium to the axon terminus. So that's what it's waiting for, is that depolarization, that action potential being sent down the axon. So that's step 2. Step 3 is once calcium goes into the actual cell space, it's going to have a protein called synaptotagmin to react with snare proteins that essentially tell the synaptic vessels to fuse with the membrane. What do I mean by that? It's going to tell the vessels that hold neurotransmitter to go to the membrane to release them. That's all it's doing. It does this through snare proteins. I haven't seen them ask about synaptotagmin. I have asked them ask about snare proteins. Snare protein is essentially the protein that the vesicle is going to use to bind to the axon terminus and then release exocytose, the neurotransmitter. So this binding to release neurotransmitter into the space, that's step 3, and it happens because of calcium. The higher the impulse frequency, so the more action potential is being sent, the more vesicles will exocytose, meaning the more neurotransmitter that's released. So if we have a stronger stimulus, we have more action potentials, we have more calcium, we have more vesicles, we have more neurotransmitter. Everything gets more. Okay. Once the neurotransmitter is released, it's going to diffuse across this tiny little space, across this, what we call the synaptic cleft, and it's going to bind to its specific receptors on the other side. So you can have multiple neurotransmitters hanging out in this axon terminal, and when they're released, there's ones that will bind specifically to their actual receptor, and that receptor will be specific for the signal that was sent. They are often chemically gated ion channels that they're binding to. Chemically gated meaning chemical binds, channel opens. It'll cause a local graded potential, a local depolarization. So our next step, step 5, or I don't know what step I'm on even, maybe 5, yeah, 5, is binding of the neurotransmitter, opening those ion channels, and will create a graded potential, a mild depolarization that, if enough binding occurs, could generate a new action potential. If not enough binding occurs for whatever reason, if maybe there wasn't that many action potentials initially sent, so not many neurotransmitters were released, and there's only a weak amount of neurotransmitters that bound, then we won't have that signal be propagated, and that's an area of control. Okay. This event could be excitatory or inhibitory. It can cause something to happen or can inhibit something. It doesn't always have to be an excitatory. And then our next step is the neurotransmitter's effects are terminated. It can be terminated by 3 ways. The neurotransmitter can undergo reuptake. It can essentially get taken back into the axon terminal and recycled. This is by astrocytes. It can be degraded by enzymes, or it can diffuse away from the synaptic cleft into the interstitial space and then be reabsorbed or broken down elsewhere. Okay. All right. So that was your neurotransmission. Big key takeaways would be calcium is the kicker, right? Influx of calcium is your big ion of control of neurotransmitter release. And then know the 3 ways that neurotransmitters can be broken down, or essentially to stop the process: reuptake, degradation, or diffusion away. Okay. When it comes to neurotransmitters themselves, there are multiple different types. My goal for you, I'm just going to go through kind of in general for the next couple minutes. I have a few minutes left before we break. My goal for you is to learn a few of them, but not to have to learn all of them. So acetylcholine would be one I would want you to commit to memory because it's the first identified, so it's the one that they know the most about, right? It's used also in the autonomic nervous system and the central nervous system, so it's widely used.
I do.
It comes from acetic acid and choline, from the enzyme choline acetyltransferase. It's broken down by acetylcholinesterase. So these would be enzymes I would also know as well. Your catecholamines would be another category I would also recommend that you look into and that you know. These are your dopamine, your norepinephrine, epinephrine. and they all come from the amino acid tyrosine. So they have the same
Break around then.
structure. That's a great biochemistry question. Okay. Great. The indolamines also are biogenic amines. They're in a different category from catecholamines. Serotonin and histamine would be the two. And I would know their precursors, tryptophan and histidine. So that's really where I find a lot of questions for neurotransmitters is either in the conversation of biochemistry, what amino acid is the precursor, in acetylcholine, potentially asking about its enzyme of creation and degradation. And then also in some instances, of course, what effects do they have in the body, but that's widespread. And we'll lean into some of those a little bit more when we get into pathology. One piece of the NPLEX that's interesting is that the NPLEX doesn't have a psych section. So you aren't going to necessarily be asked about depression, anxiety, bipolar, schizophrenia, any of those type of pieces, and the neurotransmitter relationship. So leaning into more of how do neurotransmitters affect the organ systems that you are having to learn, as much as we want to connect them to those psychiatric conditions, because that's how we learn them often. You're not tested on that on our NPLEX exam, ironically. Don't know why. Okay. I wouldn't necessarily worry about learning the amino acids role in neurotransmission, except for potentially GABA. I'm hesitating because I also glutamate and glycine I will talk about later, but this would be my third category of priority, right? If we're ranking learning these neurotransmitters, acetylcholine first, your catecholamines and indolamines next. Amino acids would be third on my list if you have brain space, but not if you don't. I wouldn't worry about your peptides, your neuropeptides as neurotransmitters. I would not worry about your purines or your gases and lipids. Okay, these are taught or they're tested on less. Endocannabinoids could come up. It seems like it's dependent on the year. I see them come up more in NPLEX 2, though, in that discussion. They are lipid soluble. They do have kind of some controls here, some supports here. They are synthesized on demand. Okay. The other piece with neurotransmitters that is helpful to understand is whether they're going to be excitatory versus inhibitory. Typically, if you're thinking about, if you're thinking about excitatory, it's going to be depolarizing. If it's inhibitory, it's going to be hyperpolarizing. So if an enzyme or a neurotransmitter is excitatory, it's going to cause depolarization. So you could think about what channels might be actually opened or closed. If it's going to be inhibitory, hyperpolarizing, it's going to cause hyperpolarization. And so you could think about then, okay, potassium channels are probably going to be most affected, or they're keeping sodium channels closed. It's one or the other. GABA and glycine are inhibitory. Glutamate is usually excitatory. Acetylcholine and norepinephrine could be both, right? So a lot of these could be either or. It depends on their receptor that they're binding to. We see that more in the autonomic nervous system. Acetylcholine is excitatory at your skeletal muscles, but it's inhibitory in cardiac muscle, right? So it's going to depend on the receptor type that it binds to. I'm just seeing what else we need to know. We already talked about this and this. This is your G proteins. Great. Awesome. All right. We'll take our break now and then we'll transition. We'll do a little bit of autonomic nervous system when we come back. And then we'll move into a little bit of GI, and then we'll round out the day with some renal and cardio at the end. So let's take a 10-minute, come back at 10 after, and then we'll finish up neuro and then go into a little bit of gut. Okay. Awesome. Yes, I'm just going to check with the online folks just to make sure, because I was hearing some sound, so I want to make sure they don't need anything from me. Yeah, I think so. Okay.